Hello, I'm Guilherme Oliveira from the Federal University of Rio. Today I'm going to talk about Cryo-EM and how we can use general transmission electron microscopy to visualize pathological protein filaments at near atomic resolution and with unprecedented level of details. Before starting my talk, I would like to share with you my the new website from our lab. So since I'm back from my postdoc in United States, I run in the structural bioengineering laboratory at the Federal University of Rio. So I invite all of you to take a look in our website and our most recent publications early this year in chemical science. So we've been working with very nice stuff in P53 and also cardiomyopathies. So in the field of structural biology and molecular biology. And also I would like to thank the people in the lab. So these are the guys doing the hard work. So I'm pretty proud of this team. So we are running in full capacity. We have more than 10 students, including postdocs and graduate students, and also undergrads. Also, a special thanks for the collaborative work with collaborators inside Brazil and also abroad. And of course, my previous mentors, my former mentors, also a special thanks. And okay, so it's also a great opportunity to emphasize the structural biology facilities we have in Rio, so this beautiful city of Rio de Janeiro, and also in the campus of the Federal University of Rio, we have a very nice technological improvements in the nuclear magnetic resonance center. So we have these very nice high field NMR spectrometers, 900 megahertz and 800 megahertz. And in a pretty close city to Rio, in São Paulo, we also have the National Laboratory for Nanotechnology, and they have very nice stuff in the field of cryo-EM. So it's a pretty exciting time to study and perform structural biology projects in our country. Okay, so since this is structural biology symposium, first of all, we should think about the definition of structural biology. And for me, it's a pretty simple explanation. So structural biology is to understand the molecular architecture of proteins and also nucleic acids. And more than that, we should think about what we can learn from a structure, right? So a molecular structure of a protein or nucleic acids. And it turns out that the structure alone, the structure itself, it's not enough to understand the function of these macromolecules. And what we can really learn from a structure is to have a more mechanistic picture of how the protein works. And to doing that, we definitely need some structure information. So if you can take a look, these cryo-EM, these 2.2 angstrom resolution cryo-EM maps it's just a cross section of the map from the beta galactosidase protein. It's a 480 kilodaltons protein, and the cryo-EM densities in these gray meshes, we can visualize them pretty clear, and it's becoming pretty evident that the cryo-EM field is pushing the resolution limits to a level, an unprecedented level of detail and resolution. So we can see, for instance, in this region, the density of catalytic water right in here. Hopefully you can see my green pointer. And also it's possible to see cofactors like the magnesium. So think about the potential we have with cryo-EM, for example, for studying structure-based drug design. So you can have right now in like a week using just in a cryo-EM lab, it's pretty easy to have several structures in like a week or in a month. Imagine you can try to form complexes of a specific target and different molecular hits. So it's a very exciting time to think about these applications in the drug design field. And what else we can learn by having structure information of molecules? So for instance, we can also have an idea of how the molecules have evolved. So to explain that, I bring a pretty nice example from a paper we did in my postdoc. So we've been working in collaboration with people in Pasteur Institute. So they have the chance to isolate from a hot acidic spring in a city called Beppu in Japan. So this is these acidic springs. They were able to isolate a primitive virus called sulfolobus polyhedrovirus 1, SPV1. So this is a frozen hydrated micrograph of this virus right here. And what is interesting, besides the 3.2 angstroms resolution map of the virus envelope, we are also able to see and capture some molecular details about the genomic DNA inside the virus. And it's interesting to explain that this icosahedrovirus, they are forming this genomic DNA in the A-form DNA instead of the B-form DNA right here. That is the most common DNA found in biology. And what is interesting to note is that the deviations in the conformation of DNA once we compare the A form and the B form, so we can see pretty easily the differences in here, and also differences in the distance we can measure. For example, the rise per base pairs and also the rise per helix turn. And from 2D classification jobs from the genomic DNA of this virus, we are able to measure these distances and have an idea that these primitive virus are really forming, are really organizing themselves in their DNA in the A form instead of the B form. And this has a lot of implications to the process of how these viruses behave in these pretty harsh and nasty environments. So the relevance of this work is to understand that several organisms living in this kind of bizarre environments, they probably use a different molecular DNA architecture to keep them alive in this kind of environment, especially because the A form DNA is also called desiccated DNA, because as you can see here, the B form has the phosphate groups located in the surface. In contrast, the B form, actually, the A form doesn't have the phosphates in the surface, so they call these the desiccated DNA. So it's pretty difficult, for example, to have water hydrogen bonding, so they can be compromised. And that's a way to explain how these viruses survive and keep their genetic information in these harsh environments. So this is another application that we can learn from structural biology to understand the conformational chains during the evolution of specific organisms. Well, and roughly speaking, so the mechanism to understand how protein works, so we should definitely have we need structural information of proteins. So by having these structural data, we can better understand how they work. So for example, these are just generic information. So by having the structure of luciferase, we can understand how these proteins produce light, so that the events of bioluminescence. And there's a lot of implications, for example, for sexual attraction, and also the camouflage for these tiny bugs, right? And it's also interesting to think about how oxygen is transported in our red blood cells by checking the complex of the heme and the oxygen inside the hemoglobin protein. And also, for example, in the case of muscle contraction, we can have a look of in the structure of the thin filaments and also the myosin filaments, and have an idea of how muscles contract. So these are pretty nice example to illustrate how the structure information is important to better understand how protein works, right? Well, but of course, life it's not that easy. So this slide it's pretty interesting for me because we try to explain an important concept. So some proteins in our body and other organisms, they undergo some weird transformation in their 3D shape, and their physiological role starts to being compromised, and they start to assume pathological roles. So, for example, this is really a threat to the organisms, because these proteins tend to form polymers inside the cells, and in most parts of the cases, these polymers becomes toxic. Not only the polymers, we mean the amyloid filaments, but also the intermediate species, the oligomers. And these kind of pathological and weird transformation of some proteins explains a lot, and is really associated with some neurodegenerative diseases like neuronal disorders that includes Parkinson's, Alzheimer's, and there are several others. And we have a bunch of these proteins. So the prion, it's a nice example, alpha-synuclein, and so on. And the other point in here is that we also have the story of the functional amyloids. So the functional amyloids, I would say this is a good explanation, a good representation of how a functional amyloid would be. So it seems like bad, but it's not really bad. So the point in here is that these functional amyloids can also polymerize, form these nanofilaments, and also form these beta cross sheets. But they are really doing a nice work for the cells, and they have physiological role. They don't have this pathological role. And I think this is a cool slide to represent the concept, to introduce actually the concept of the amyloidogenic proteins. So these are proteins undergoing some specific transformations and becoming very toxic to the cell. And well, one of the hot topics in the literature nowadays is to understanding in which circumstances these kinds of pathological filaments, the amyloid filaments, and they are formed. So the conversion, what are the circumstances that leads to the pathological conversion? And it's mostly related to neurodegenerative disorders, but there's some recent data from our group and a few others in the world showing that there's not an exclusive way to explain neurodegenerative disorders, but there's also other proliferative disorders like cancer that can also form this kind of material, this pathogenic amyloid material. And in the case of cancer, we have this nice story on P53. So it's like 20 years of research on that. So Professor Gerson will give a lecture on that. But just to illustrate, so we've been trying to understand what would be the process of aggregation in a specific segment of the DNA binding domain in P53, specifically the amyloidogenic peptide that is shown in here. So by using a molecular dynamics simulation and also some wet lab experiments with transmission electron microscopy, we are able to capture how would be the conversion process in the early stage of aggregation, the mid stage, and also the final stage. I mean, when you have these major fibers. So what we first discovered is that once we have this peptide forming the dimers, you can have a mixture of beta strands that forms parallel and anti-parallel organization, like as shown in here and in here. And this is the major, distribution we have. And more interesting than that is that you have this hydrogen bonding that is formed in the main chain, and also in the side chain and side chain interactions. And this is also cool because we can try to understand the strand orientation during this nucleation process. And also when we keep going in the MD simulations, we also have information for the mid stage, so we can form these hexamers of the amyloid peptide in the form of a sandwich like beta sheets association, and it's pretty heterogeneous, right? And moving to the wet lab, we can also grow in vitro some conditions with these peptides and form these pretty nice amyloid filaments. And they are major fibers. We can see that they have some polymorphism at the quaternary structure level. So the width of these filaments, they are quite, there's a broad distribution of them. So we can measure that. So these are micrographs with unprecedented level of detail. So it's pretty nice to see how the amyloidogenic peptide of p53 can form these weird fibrils, right? And what's the relevance of this work at all? It's just to show that probably the process of converting the p53 to a functional and like monomeric protein to a amyloidogenic and pathological protein, it's probably polymorphic, not only in the final stage in the major fibers, but probably right in the early stage you can also have some polymorphism, for example with these parallel and anti-parallel beta sheets. So it's pretty cool for us to understand the mechanism of conversion. And since we are talking about polymorphism, we can move back to the neurodegenerative disorder with the example in here. So this is another work I did during the postdoc. So getting back to alpha-synuclein involved in Parkinson's disease. So we are able in this work to play with 4 different constructs of the full-length alpha-synuclein and some familial forms mutations like the A30P, the E46K, also the A53C, and we generate in vitro amyloid fibers from these different mutations, and we are able to check how polymorphic they are, right? So that's pretty nice as well. And what we can, these are frozen hydrated micrographs. So what we can do with that, so we can extract these filaments, we can align the filaments, and from the aligned filaments, we generate a power spectrum that is just the Fourier transform of the real space micrograph, and we get something like this. And from these power spectra, we can extract some important values, like distance values from the fiber that give us clear evidence on how different they are, right? So for instance, we can measure the pit distance. So filaments, these amyloid filaments, they have helical symmetry. So we can measure the pitch distance that is shown here. And what we found is that the wild type and the A53T major fiber, they have similar pitches. So it means a similar pitch. So it means they seems to be pretty similar in terms of their final product, but they have different kinetic traces as shown in here once we record them by thioflavin T. And that's important because, and pretty interesting as well, because we can show that probably the formation of the oligomers, the intermediate species, are quite different and polymorphic at these early stages of aggregation, and that what give rise to these pretty high increments in thioflavin T. during these traces. And when compared with the other mutations like the A30P and the E46K, they are drastically different fibers. So for instance, the A30P has a 0.4 nanometer pitch, and the A46K has like 1.4 nanometers pitch. So this is clear information for us that the fibers formed by these familial mutations, they really produce like polymorphic amyloid fibers in alpha-synuclein. And it is also a cool example to show that cryo-EM, it's not only used for example, to determine the near atomic resolution of structures. It's also important, for instance, to get some measurement information, like the pitch distances in this kind of approach, the helical filaments. And it's also true that the polymorphism in fibrous, they are not only present once you have mutations, but you also have polymorphism inside the same polypeptide chain. So the wild type full-length alpha-synuclein can be polymorphic as well. So this is other nice example from another paper showing that you can isolate and determine the cryo-EM near atomic resolution of a rod polymorph and a twist polymorph. You can see they have different pitch. So this is a longer pitch when comparing the twist polymorph. There's a shorter pitch in here. They also assume different shapes, the protofilaments. So this is one protofilament, this is the other one. So this you have in this one, you have an omega shape, and in this one you have an L shape, as shown in here. It's also pretty cool to see the amino acids involved in the protofilament interface. You have short side chains in here. Some of them are mutation sites, like histidine 50, the A53, and by checking these two filaments, it's pretty nice to see how diverse the arrangements. Because, for instance, in this case, in the rod polymorph, you have the mutation sites, the common mutation sites located inside the protofilament interface. Once in the twist polymorph, you have the mutation sites, the most common mutation sites located in the outer surface. So it's pretty nice to see how diverse this polypeptide chain can be, right? We also have some ideas on the distortion side. So the polypeptides pre-distorted, right, in these regions, and we have like short side chains in these strong distortions in here, like glycine residues, valine residues, alanine residues. And the core, it's also very pretty much hydrophobic. And we can see, so again, what we can really learn from checking and having this kind of detail, this near atomic resolution detail on different polymorphs. So we can try to figure out the forces that hold the filaments together, right? And it's also important to mention that the Parkinson's disease is just one type of neurodegenerative disorder in which alpha-synuclein is involved, but there's a lot of other synucleinopathies, and some of them you can try to understand you can try to correlate a specific polymorph to a specific clinical outcome. So it would be nice, for instance, to understand how a specific polymorph give rise to a specific clinical outcome from a specific disease. So that's the reason why I consider structural biology pretty important in this kind of approach, in this kind of evaluation, and in this kind of level of detail. So I think it's much exciting to see these near atomic resolution structures in a video. So this is pretty challenge for me, but let's try to play at the same time both movies, so we can see both polymorphs, the the rods in blue and the twist in orange. So here you can see the cryo-EM densities for both amyloid filaments, the atomic model that you can build from the cryo-EM maps. And okay, maybe I should stop the video right now to show the side chains right in here, right in here. Okay, so let's play them individually now. So first in the left side of your screen, let's see some of the details I mentioned before. So this is pretty cool because we can see each of the protomers. We have 5 protomers in here, and you can see these some details. For example, there's a ion pair right in here. So this is lysine 80, this is E46. So it's forming an ion pair, salt bridge all over the axis of the fibro. Most of the residues in here are short side chains and hydrophobic. There is a surface, there is charge, outer surface of the fiber that is pretty much charged residues. So this is pretty cool. It's a high level of detail, and we can understand better how the fibro is packed together with this level of information. And also the L-shaped polymorph, the twist polymorph, we have some differences. So it's an L shape, it's not an omega shape. You also have a charged residues outer surface in here. Now the mutation sites, they are not located in the interface of the protofilaments. It's pretty much hydrophobic, the interface. You also have the charged residues in these parts. And also the mutation sites, the common mutation site, histidine 50, A53, it's located in the outer side of the fiber core. So it's pretty much to understand these filaments. And it's also important to mention and point out that cryo-EM is passing through a revolution, and it's becoming tremendously popular. Solving the structure of different amyloid filaments. So this is just an explanation, and there's a lot of examples nowadays of cryo-EM filaments that were solved by cryo-EM. So we have different polymorphs, for example, from tau related with different disease, the A-beta involved in Alzheimer's disease, the prion protein as well. It's pretty interesting. This is another example for an A-beta 42 filament. So it's very nice to understand this type of fibers, because the 2 protofilaments, the left one and the right one, they are held together by an ion pair, this salt bridging here. So you can see in this video the densities. And it's also nice to see that the fibros, the 2 protomers, the 2 protofilaments, this one protomer, this is the other protomer, they are staggered, one from the other, and it's pretty clear in the last frame of this video. So the salt bridge is forming all over the axis. So we can imagine if you want to dissociate these filaments, these protofilaments, you can just play with, for example, ionic strands. So you can see that the salt bridge is not forming like this in this distance. They are forming with this protomer in here. So this is the density for the lysine, and this is the other density forming the ion pair. It's all over the perpendicular to the filament axis, though. So it's pretty cool to have this type of detail and understanding the forces behind the filaments, so the force that keeps the filament together. And there's also a nice story about these challenging oligomers. So we have information that they could be the building blocks to polymerize the fibrils. We can discriminate small oligomers, the large oligomers during the polymerization reaction. In the course of the polymerization reactions recorded by thiophanate T, we have this stepwise aggregation process of alpha-synuclein, the full-length alpha-synuclein. So this is also a pretty nice work. They are publishing communications biology. So I invite you to take a look if you're interested in this team, in this thematic. And I guess that's all that I have to show. So I'm running out of time. So just to acknowledge the funding. So especially thanks to FAPERJ, that's our funding agency in the state of Rio, and also the Pew Charitable Trust Foundation. And I would like to thank for listening. I hope I can have your questions later on today. And thank you all. Bye-bye.